constantly throughout the body through a process known as cell division. When a cell becomes old or damaged, the body sends signals to the cell to die and be replaced with new cells in a process called apoptosis.
Sometimes this process is disrupted and old or abnormal cells continue to divide. This is known as a cancer cell. Cancer cells do not perform normal cell functions, like apoptosis, meaning they continue to divide and spread throughout the body. When a collection of cancer cells form, the grouping of cells is called a tumor.
Tumors are either known as cancerous or benign. When cancerous tumors spread throughout the body, it is known as metastasis. Although benign tumors are known as metastatic, they can still be harmful to your body if not removed.
Cancer cells are so dangerous due to their special abnormalities. They are able to conceal themselves from the immune system so they are not destroyed. Blood vessels grow around tumors so they are continually provided with blood and nutrients, and they are able to continue growing and dividing, having many changes in chromosomes, sometimes even double the number of chromosomes compared to normal cells.
Cancer cells are developed by gene mutation, a change in one or more genes. Genetic changes include errors as the cells divide, damaged DNA, which is harmed by the environment, or carcinogens or toxins found in the environment that promote cancer cell growth, some including tobacco, UV rays, and other chemicals.
Doctors and scientists work tirelessly to find cures for cancer. One of these therapies is something called gene therapy. Gene therapy replaces a faulty gene or adds a new one in order to cure a disease or strengthen the immune system and the body's ability to fight the disease. Genes can either be turned off, which in turn no longer promotes disease, or turned on, healthy genes which make disease cells easier for the immune system to attack.
In some scenarios, the body's immune system does not recognize disease cells as intruders. Gene therapy can train the immune system to recognize cells that are threats through the delivery of genetic material. This material is delivered using a carrier or vector. Vectors, with genetic information they have, can target the cause of disease and change the way a cell functions. They're like vehicles designed to transfer therapeutic genetic material, such as an active gene, directly into a cell.
Vectors are taken from viruses because viruses are able to efficiently and easily find their way into cells. In order to make vectors safe to use for the transfer of therapeutic genetic material, all viruses are removed. Once the virus is removed from the vector, it is injected into the patient's body through a vein or into the tissue, where the cell then absorbs the vector carrying the altered genes.
One of the diseases gene therapy is used to treat is cancer, where we use the same basic principles of gene therapy to stop abnormal cell division in cancer cells and train the immune system to recognize and kill cancer. Gene therapy is an alternative or additional treatment intended to replace current practices like chemotherapy.
Oftentimes, patients see better results with both chemotherapy and gene therapy treatments. But the goal for the future of gene therapy is to replace radiation and chemotherapy due to their harsh side effects. The systemic toxicity of chemotherapy includes many short-term and long-term effects, including the risk of developing other types of cancer. So developments in gene therapy are crucial for the future of cancer treatment.
Gene therapy includes a wide range of treatments, such as immunotherapy. Immunotherapy is a type of gene therapy that involves genetically modifying patients' immune cells to treat their cancer. It has become the fifth pillar of cancer treatment.
Immunotherapy strengthens the patient's immune system to attack tumors. It includes many forms of treatments, including cancer vaccines, which destroy cancer cells with viruses that perform cell lysis, which is the disintegration of the cell wall or membrane. Immunotherapy also helps to decrease tumor blood supply and restore normal functions to cancer cells.
Oncolytic agents are a form of gene therapy where viruses are genetically modified to target or destroy cancer cells while not harming the body. Both colon and bladder cancer have shown survival benefits and reduced metastasis using oncolytic viral agents. In the study Onyx 015 viral therapy, an adenovirus vector was used to limit a protein in cancer cells, which allowed for rapid cell division. This type of gene therapy has seen best results when combined with chemotherapy.
Gene transfer includes introducing a foreign gene to cancer cells, including genes that cause cellular death when expressed. The most commonly used viral vector, adenovirus, which causes cells to lack ability to replicate. The success of this therapy was seen in tumors of the lung, prostate, and pancreas, which treated with success in animals. Gene transfer increases effectivity of chemotherapy treatments and is also known as the only gene therapy product to have global regulation approval.
In 2013, a new gene editing tool called CRISPR, which stands for Clustered Regularly Interspersed Short Palindromic Repeats, was found. CRISPR proved to alter the DNA of human cells easily and quickly, while many other forms of gene therapy weren't able to do so. Some scientists said that using CRISPR was the equivalent of using a pair of scissors. Other scientists said it was so easy to use that a high schooler could do it. It gained popularity for its simplicity and its availability to places across the world than other gene editing tools.
CRISPR was inspired by nature. The idea came from a defense mechanism found in microbes and bacteria. The idea is to protect themselves against invaders like viruses. These microbes capture fragments of the intruder's DNA and hold them away as segments called CRISPRs. If that same invader tries to attack again, those stored DNA segments assist an enzyme called Cas9 to track down and slice up the invader's DNA. Once scientists discovered how much potential CRISPRs had, many labs successfully adapted the system to edit any DNA sections. CRISPR works by using two main components, a guide RNA and a DNA cutting enzyme called Cas9. Scientists designed the guide RNA to mirror the DNA of the gene to be edited. RNA and Cas9 link up with each other and it leads the Cas9 to the gene that needs to be edited, also known as the target. When the guide RNA matches up with the target gene's DNA, Cas9 cuts out the DNA that's being edited. CRISPR is used in different ways. In some cases, the target gene DNA is scrambled while it's being repaired, and in other cases, scientists can manipulate genes by adding in a new segment or editing single DNA letters.
CRISPR is customizable, meaning it's able to edit any DNA segment within 3 billion letters of the human genome, and it can also be easily upscaled, or can use hundreds of RNA guides at a time and evaluate hundreds or thousands of genes at a time.
However, these are some of the limitations of CRISPR. Sometimes it cuts DNA outside of the target gene, what's known as off-target editing. In other cases, it starts breaking random parts of the genome. The cell can start binding things together in odd ways, and there's some concern about that becoming cancer. Another thing is getting CRISPR components into cells, co-opting a virus to do the job instead of ferrying genes that cause disease. The virus is modified to carry genes for the guide RNA and Cas. And lastly, the virus that carry CRISPR can infect multiple types of cells. For example, editing muscle cells when you want to edit liver cells.
CAR T cells are another type of gene therapy that were found and have been used since 2017. 6 CAR T cell therapies have been approved by the FDA, and all 6 of these CAR T cells treat blood cancers such as lymphomas, some forms of leukemia, and multiple myeloma. CAR T cells are associated with being a living drug. They are made by collecting a sample of T cells, or a type of white blood cell called lymphocytes, from a patient and genetically modifying to produce proteins on their surfaces called chimeric antigen receptors, aka CARs. The CARs recognize and bind to specific proteins or antigens on the surface of cancer cells. Every patient has specific CAR T cells that are customized for every patient. We collect samples, re-engineer, grow many, and newly engineered cells infuse into the patient.
It has been proven time and time again that CAR T cells kill cancer cells. However, there are still many downfalls to this type of treatment. Cytokine release syndrome, which is when T cells release cytokines, which are chemical messengers that help stimulate and direct the immune response. CARs and infused T cells flood the bloodstream with cytokine, causing serious side effects, like high fevers and blood pressure dropping, and it can be fatal. Also, neurological effects, for example, severe confusion, seizure-like activity, and impaired speech, and still much more to research as the neurological effects are still unclear. Another new idea has evolved from further research of CAR T cells. The idea is using collected T cells from healthy donors and would be able to use them immediately rather than having to be manufactured for each patient. This would create CAR T cells to be considered off the shelf, and more patients could have access to this new treatment. Off-the-shelf CAR T cells are now being tested in small clinical trials. Gene editing technologies like TALON and CRISPR are being used to induce the donated T cells to produce CAR T cells. There are also other research groups that are using nanotechnology and mRNA-based techniques that allow CAR T cells to be created inside the body.
Promoter of the gene. Enzymes are used to target the RNA during transcription, especially during the elongation step, which can be inhibited through transcription factors. Translation is the process of protein synthesis according to the mRNA template strand, which includes tRNA. On the second step of translation, tRNA is targeted for inhibition, which blocks the protein synthesis process.
Oncogenes are targeted by gene therapy, as they are genes that can cause cancer once mutated or when expressed at high levels. Many oncogenic pathways lead to altered transcription or translation of various proteins. Scientists can manipulate these genes in gene therapy.
Cancer treatments are an ever-evolving project that scientists continue to work on. As uncovered in this video, gene therapy is a newly developing cancer treatment that is customizable for each patient that suffers from this fatal disease. It is crucial that we continue research into this treatment type as it has unforeseeable potential for the future of cancer treatment.
Thanks for watching, Mr. Wang!